Imagine having a kind of secret instruction manual inside every one of your cells, like a super secret blueprint that dictates, well, everything about you, from the color of your eyes to how your cells fight off disease. What if we told you we're diving deep into that blueprint today?
Yeah, and what's truly fascinating here is just how much our understanding of this blueprint, the gene, has evolved. I mean, from those early sort of abstract units of inheritance
Right.
to the complex molecular machines we know today, it's really been a journey of incredible scientific discovery,
Uh-huh.
and it's still unfolding.
Absolutely. Today, we're taking that deep dive right into the very core of molecular biology. We're drawing insights directly from a really comprehensive source, Molecular Biology, the Gene 7, 2013. Our mission for you is, well, to give you a shortcut to being well-informed about this hidden world of genes. We want to reveal some surprising facts, those genuine aha moments that might fundamentally change how you see life itself. So, to set the stage, let's go way back to the foundational understanding of heredity. Think about Gregor Mendel's groundbreaking work.
The pea plants guy.
Exactly. He basically showed us how traits are passed down, leading to predictable ratios. Like in pea plants, you might see that 3.1 ratio of dominant to recessive phenotypes.
Right.
But what's truly remarkable is that his laws didn't even depend on dominance to work.
Yeah.
You see this clearly in snapdragons, right? A cross between red and white gives you pink.
The intermediate phenotype.
Yeah, and then the next generation shows a precise 1.2.1 ratio of red, pink, and white. So he was demonstrating these fundamental principles as independent segregation without even knowing what a gene was. It's almost like understanding gravity before knowing about mass.
Precisely. So Mendel gave us the rules, the sort of abstract rules of inheritance, but the actual molecular players. Still a complete mystery.
Right.
And this is where the revolutionary idea came in. Linking these abstract units to physical structures inside the cell, specifically chromosomes. Thomas Hunt Morgan, he started his pioneering work with the tiny fruit fly, Drosophila melanogaster, back in 1908. And he made this striking discovery when a white-eyed mutant just appeared spontaneously.
Wow, just like that.
Just like that. And this seemingly small event, it opened the door to complex linkage studies. By 1915, his group had mapped over 85 mutant genes on Drosophila chromosomes.
85.
Yeah. And they revealed this strictly linear arrangement. This linearity, if we connect this to the bigger picture, this linearity was a powerful early clue to the gene's physical nature. It suggested they weren't just abstract ideas, but had a defined place, like beads on a string.
Okay, that makes sense. But for years after that, the big mystery remained. What was a gene chemically made of? I mean, for decades, right up until the mid-1940s, the prevailing scientific view was that genes had to be proteins.
Right. The complexity argument.
Exactly. They just seemed complex enough to convey all that genetic information, didn't they?
Yeah.
DNA was known, sure. It was a unique molecular constituent of chromosomes, but it was generally assumed to be merely a molecular scaffold.
A framework.
Yeah, a scaffold for these still undiscovered gene-carrying proteins. And here's where it gets really interesting, because the truth turned out to be quite different.
Indeed it did. The definitive experiment, the one that completely shifted scientific understanding, came in 1944. Oswald T. Avery and his colleagues made what was truly a momentous announcement.
Okay, what was it?
DNA, not protein, is the active genetic principle. Their critical evidence was, well, profound. They showed that the transforming activity in pneumococcus bacteria, the ability to pass on genetic traits, was completely destroyed by deoxyribonuclease, an enzyme that specifically degrades DNA.
Okay, so targeting the DNA killed the transformation.
Exactly. And crucially, this activity was not destroyed by ribonuclease, which degrades RNA, or by various proteolytic enzymes that break down proteins. It was a clear, undeniable signal. DNA held the key. It essentially redefined the very substance of life's blueprint.
Wow. So once we knew DNA was the blueprint, the race was on to figure out its iconic shape, how it's actually built. And it was a true global scientific effort.
It really was.
Early X-ray diffraction patterns were actually taken way back in 1938 by William Asbury. But it was the high-quality X-ray diffraction photos taken by Maurice Wilkins and Rosalind Franklin in the early 1950s that truly pushed things forward.
Those are key images.
Yeah, their images strongly suggested a helical structure made of more than one polynucleotide chain. Then, in 1952, Alexander Todd's group made the crucial finding that phosphodiester bonds regularly link nucleotides in DNA, giving it that stable backbone.
Putting the pieces together.
Right. But it all culminated in the monumental work of Watson and Crick in 1953, who famously proposed the double helix model. Two polynucleotide chains twisted around each other, the bases projecting inward, forming those complementary pairs.
The A with T, C with G.
Precisely. And a critical piece of their puzzle was Chargaff's rules, that observation that A always pairs with T and C always pairs with G. It was a massive aha moment, wasn't it? It truly changed everything, revealing how the blueprint could store and, crucially, copy information.
And with the structure known, the next logical step was understanding how this ingenious blueprint copies itself. The Messelson and Stahl experiments, they elegantly showed that DNA replication is a semi-conservative process.
Semi-conservative.
Meaning each new double helix retains one parental strand. It's a beautifully efficient mechanism for faithful copying.
Okay.
And the remarkable enzymes that catalyze this DNA synthesis are called DNA polymerases. What's fascinating is they use a single active site to add any of the four building blocks, sort of adapting to the incoming nucleotide.
But wait, if it's one active site, how does it get it right so often? How do they maintain such incredible accuracy?
That's a great question. They employ something called kinetic proofreading. They dramatically slow down the addition of incorrect nucleotides by as much as 10,000-fold. It makes adding the wrong piece incredibly difficult.
10,000 times slower. Wow.
Yeah. And to boost their efficiency, make sure they don't just fall off the DNA strand, these polymerases get help from sliding DNA clamps.
Sliding clamps.
Yeah, they're like these donut-shaped proteins that encircle the DNA. They bind tightly to the polymerases and greatly increase their processivity.
Processivity?
Yeah.
Meaning they stay on the job longer.
Exactly. They can add many, many nucleotides without dissociating. Now, in our linear eukaryotic chromosomes, there's also this thing called the end replication problem.
Right, because you can't fully copy the very ends, can you?
Correct. DNA could be lost with each division. But life found an elegant solution through telomeres, these specialized ends, and the enzyme telomerase, which helps maintain them. It ensures genetic information isn't lost generation after generation.
Okay, so we've got the blueprint, DNA, and how it's copied. But the DNA itself isn't making proteins directly, right? That led to the next big concept, the flow of genetic information, famously known as the central dogma.
Right, DNA to RNA to protein.
Yeah, it became clear the DNA double helix itself couldn't be the direct template for protein synthesis, especially in eukaryotes, like us, where DNA is locked away in the nucleus, but proteins are made out in the cytoplasm.
The location issue.
Exactly. So attention shifted to RNA, the second information-containing molecule. The idea was, well, RNA gets its genetic specificity from DNA, then moves out to the cytoplasm to serve as the template for protein synthesis.
The messenger.
Like DNA holds the master copy in the library, but RNA creates the working copies to take out to the factory floor.
Exactly. And the first step in that expression process is transcription copying DNA into RNA. This process is primarily catalyzed by RNA polymerase.
Okay.
Interestingly, bacterial RNA polymerase is a single enzyme, and it uses a specific part called a sigma subunit to recognize promoters, which are basically the start signals for genes.
But it's different in eukaryotes.
Yeah, in eukaryotes, like our cells, we have 3 different RNA polymerases. Pol2, for example, transcribes most protein-encoding genes. And one fascinating detail is how the TATA-binding protein, or TBP, think of it as a key door opener for many genes, works.
The TATA box, right.
Exactly. When TBP binds to that specific TATA sequence in eukaryotic promoters, it causes a dramatic 80-degree bend in the DNA.
It bends the DNA.
Yeah. This structural change actively helps drive transcription initiation. It's quite clever. And then there's the challenge of nucleosomes in eukaryotes.
Ah, the DNA wrapped around proteins.
Right. Think of your DNA not as a loose string, but expertly wound around these tiny protein spools called histones, forming structures called nucleosomes. It helps organize things, but it's also a barrier.
To the polymerase.
Exactly. How does it read the DNA when it's schooled up like that? Well, there's a protein complex called FCT. It basically helps by temporarily unspooling the DNA from these histone reels, clearing a path for RNA polymerase, and then quickly respooling it afterwards.
Like a tiny molecular stage crew.
Yeah.
That's incredible. So it's not just copying the message, it's refining it. Once RNA is made, especially in eukaryotes, it undergoes pretty expensive processing to become mature, right?
Absolutely. These processing events are really unique.
Like the 5-foot capping.
Right. The 5-foot capping adds a special tag, kind of like a protective helmet, and it helps the ribosome recognize the message later. Then there's splicing.
Removing the introns, the non-coding bits.
Exactly. Those non-coding intron sections of, you could almost say, genetic junk are removed, and the truly coding exons are precisely joined together. And finally, there's 3' polyadenylation.
Adding the poly A tail.
Yeah, a string of A's, which adds stability and helps with export from the nucleus. What's truly wild is the vast variability of introns in human genes. Some have just one, but the Titan gene has as many as 363.
363 introns. That's amazing.
It is. But perhaps the most surprising discovery in this area was ribozymes.
RNA acting like enzymes.
Precisely. RNA molecules that possess catalytic activity just like proteins. Examples include self-splicing group 1 and group 2 introns. They cut themselves out. And even the peptidyltransferase activity within the ribosome itself, the machine that makes proteins, is catalyzed by RNA.
So what does this all mean? RNA isn't just a passive messenger, is it?
Not at all. It's a vital, active participant, a true worker in the cell's machinery. It truly challenges that old idea that only proteins could be enzymes.
That's right. And all this genetic information, all this processing, eventually leads to its final grand performance, translation, building proteins from RNA instructions.
And the cracking of the genetic code itself was a monumental achievement. Marshall Nierenberg and Heinrich Mathai's 1961 experiment showing that poly-U leads to polyphenylalanine. That was the first piece of the puzzle.
Just using a simple repeating sequence.
Yeah. And then Hargobind Khurana's work with more complex polynucleotides further refined our understanding, revealing the entire code. Then there's the clever wobble hypothesis.
Wobble. What's that?
It explains how a single tRNA molecule, the adapter molecule, can recognize multiple codons, the 3-letter code words in RNA. It's due to flexible pairing at the 3rd position of the codon. It makes the system incredibly efficient.
Ah, okay. Saves needing a unique tRNA for every single codon.
Exactly. Transfer RNAs, or tRNAs, are those crucial adapters between codons and amino acids, each ending with a conserved 5-CCA3 sequence. And the aminoacyl tRNA synthetases are the enzymes that accurately charge these tRNAs with the correct amino acid.
They have to be accurate.
Extreme. Some even use an editing pocket for incredibly high accuracy, like a double-check mechanism. All of this happens on the ribosome, that complex molecular machinery with 3 tRNA binding sites, the A, P, and E sites where protein synthesis occurs.
The protein factory.
Right. And get this. The peptidyltransferase reaction, the actual forming of peptide bonds to link amino acids together, is primarily catalyzed by the ribosomal RNA component, not a protein.
So the ribosome itself is a giant ribozyme.
It really is. It showcases RNA's remarkable versatility and its deep, fundamental role in life.
Okay, so we've built the blueprint, copied it, used it to create proteins.
Yeah.
But what happens when the blueprint itself gets damaged? Because, I mean, in the chaotic world of the cell, perfection seems impossible, right?
That's a crucial point. The cell faces a constant challenge maintaining the integrity of its genetic blueprint. Mutations are, unfortunately, unavoidable.
Why is that?
Well, there are inherent inaccuracies in DNA replication itself, like something called tautomerization, where the chemical structure of a base momentarily shifts, leading to a misbearing.
Okay, so random chemical shifts.
Right. Then there's constant chemical damage from the environment.
Yeah.
Things like deamination of cytosine, turning it into uracil, alkylation, oxidation, just the wear and tear of being in a cell.
And didn't you mention jumping genes?
Yes. Transposons, these can disruptively move and insert themselves into new locations, potentially damaging genes. So life maintains this sort of happy balance.
Between what?
Between mutation, which actually drives evolution and allows for adaptation.
Ah, so some mutation is good.
In the long run, for a population, yes. But for the individual cell, it needs DNA repair to prevent catastrophic damage. It's a constant balancing act.
So what are these defense systems like?
They're incredibly sophisticated. Take mismatch repair. This is crucial, especially well studied in E. coli. It detects distortions caused by incorrect base pairs, like a bump in the helix.
How does it know which strand is wrong?
Cleverly. It distinguishes the newly synthesized strand from the parental strand, often by its methylation status chemical tags on the DNA, to ensure the right correction is made.
Smart. Yeah.
Then there are direct reversal repair mechanisms. These are less common but very direct, like DNA photolysis using light energy to reverse UV-induced thymine dimers.
Damage from sunlight.
Exactly.
Good.
Or methyl transfers directly removing methyl groups. But these can be costly because the enzyme itself is consumed in the process.
Okay.
More common is base excision repair. Here, enzymes called glycosylases recognize specific damaged bases and literally flip them out of the helix for removal and replacement.
Flips them out. Wow.
Yeah, and finally, when the worst happens, a double-strand break, DSB, where the DNA backbone is broken in both strands, that's serious damage.
Yeah, that sounds bad.
It is. Here, you have two main strategies. First, homologous recombination, or HR. This is the perfect repair mechanism. It uses a homologous sequence, usually from the sister chromatid right after DNA replication, as a template to accurately repair the break. It involves proteins like RAD51 and forms these temporary structures called holiday junctions.
So it uses an undamaged copy as a guide.
Precisely. If we connect this to the bigger picture, HR is like having an exact, perfectly matched spare part for a critical piece of machinery.
Okay, that sounds ideal. What's the other way?
The other main way is non-homologous end joining, or NHEJ. This is more of a quick and dirty repair. It basically just sticks the broken ends back together.
Doesn't sound very accurate.
It's not always. It's effective for speed, gets the chromosome back together quickly, but it can sometimes lead to small insertions or deletions at the break site. It represents a trade-off, speed versus perfect accuracy.
So life isn't just about perfect copying. It's about having this incredible toolkit for fixing mistakes.
Absolutely. It's a constant battle against damage. And without these repair systems, complex life just wouldn't exist.
That's staggering. So beyond just fixing errors, the cell is also brilliant at orchestrating gene expression, the whole intricate world of regulation. How does it know when to use which part of the blueprint?
Regulation. That's key. In prokaryotes, like bacteria, we see incredible efficiency. Take the famous lac operon in E. coli.
Dealing with lactose, right?
Exactly. It's a classic example of how a cell precisely controls the genes for lactose metabolism. It turns them on only when lactose is available and, crucially, when glucose, the preferred energy source, is absent.
So it checks both conditions.
Yes. It involves this clever interplay of an activator protein, CAP, and a repressor protein, the lac repressor. And both are controlled allosterically. Their shape and activity are changed when they bind to specific signal molecules like lactose or a molecule related to glucose levels.
Wow, that's sophisticated logic.
It really is. Another fascinating case is the lambdaphage, a virus that infects bacteria. It has this life or death decision.
Whether to kill the host or lie low.
Exactly. Enter a terelik cycle, destroying the host, or a lysogenic cycle, integrating into the host genome. This switch is controlled by a delicate balance between two key proteins, repressor and CRO, competing for binding sites on the DNA, and they show cooperative binding.
Meaning one helps the next one bind?
Yes, binding of one protein makes it easier for others to bind nearby. It makes the switch very sharp, very decisive. Another protein, CII, is kind of the conductor orchestrating this switch based on the cell's conditions.
And regulation isn't just about starting transcription, is it?
Good point. There's also anti-termination. Proteins like N and Q in lambdaphage can actually allow RNA polymerase to read right through termination signals, extending the transcripts. It's another layer of control.
Okay, so prokaryotes are efficient. What about eukaryotes? Like us, more complex.
Oh, yes. We see added layers of complexity. Unlike in bacteria, eukaryotic activators and repressors rarely interact directly with RNA polymerase. Instead, they act as recruiters.
Recruiters? For what?
They bring in entire protein complexes. Things like the mediator complex, which acts as a bridge to the polymerase, or those nucleosome modifiers and remodelers we talked about, like FACST helping to clear the histones.
So they call in a support crew.
Exactly. Then there are enhancers. These are DNA sequences that can be located quite far away from the gene they regulate, upstream or downstream, even within introns.
Far away. How do they work then?
The DNA loops around, bringing the enhancer close to the promoter region. They often form structures called enhanceosomes by bringing multiple activator proteins together in a highly cooperative way. The beta interferon gene is a classic example. They act like powerful boosters for gene expression.
Wow. And what about this epigenetic stuff? Changes not in the DNA sequence.
Ah, yes, epigenetic regulation. This is a huge and fascinating area. It refers to heritable changes in gene expression that are not due to alterations in the DNA sequence itself.
So how does that work?
Several ways. There's gene silencing, where large regions of DNA, like the tightly packed heterochromatin found at telomeres and centromeres, are basically shut down. Proteins like HP1 and SIR proteins are involved in maintaining this silent state.
Okay, packing it away tightly.
Right. Then there are specific chemical marks, things like acetylation, methylation, phosphorylation, that get added to the tails of histone proteins. This forms a kind of histone code.
A code on the packaging.
Exactly. This code dictates whether the genes in that region are likely to be active or repressed. It's incredibly dynamic. There's also DNA methylation, a direct chemical modification of DNA bases, usually cytosine. This also influences gene expression.
And that's linked to diseases.
Yes. For example, its role is highlighted in disorders like Rett syndrome, which involves a repressor protein called MESI-P2 that specifically binds to methylated DNA. And for a truly dramatic example of epigenetics, think about X inactivation in female mammals.
Where one X chromosome is shut down.
Precisely. One entire X chromosome is silenced, largely mediated by a long, non-coding RNA called ZIST. It literally coats the chromosome to shut it down, ensuring females don't have a double dose of X-linked genes compared to males. It's a remarkable feat of large-scale gene regulation.
That is astonishing. So much control happening around the DNA sequence itself. And that brings us nicely to what's been called the RNA revolution, right? The realization that RNAs do way more than just carry messages.
Absolutely. They're active regulators, even catalysts.
Like attenuation in the TREP operon in bacteria.
Yes, that's a great example. Ribosomes stalling on a small leader peptide region of the mRNA actually causes the mRNA downstream to fold into a specific structure. This structure prevents transcription termination, so the operon stays on when tryptophan is low. Its regulation directly linked to the act of translation itself.
Linking synthesis to need. Clever. What else?
Well, consider the CRISPR-Cas system in bacteria and archaea. It's basically their adaptive immune system.
Immune system in bacteria.
Yeah. It incorporates small snippets of foreign DNA, maybe from an invading virus or plasmid, into its own genome as spacer sequences. These are then transcribed into small RNAs.
Oh, yeah.
These small RNAs then guide CRISPR-associated protein complexes, the Cas proteins, to find and destroy any matching invading nucleic acids, DNA, or RNA. It's molecular memory and defense.
What's fascinating here is how similar the fundamental logic, recognizing and destroying foreign invaders based on memory, is to our own sophisticated adaptive immune systems, just using RNA as the key recognition molecule.
It's a beautiful example of convergent evolution, perhaps. And then, of course, there's RNA interference, or RNAi.
That was a huge discovery, wasn't it, in worms?
Yes, groundbreaking work in C. elegans, the nematode worm. They found that introducing double-stranded RNA could effectively silence genes with a matching sequence. This dsRNA gets processed by an enzyme called dicer into tiny bits, siRNAs or miRNAs.
And these silence the gene.
Yes. They guide protein complexes to either degrade the target mRNA or block its translation. RNAi has become an incredibly powerful tool for researchers across many organisms to deliberately turn down or turn off specific genes and study their function. It really cemented RNA's role as a major player in gene regulation.
So the RNA revolution isn't just about finding more jobs for RNA. It's really a fundamental shift in understanding its versatility and importance.
Definitely. And all these intricate molecular mechanisms, they're all part of life's grand design principles. We see them clearly in development, evolution, and now systems biology.
Right, looking at the blueprint in action, building a whole organism from just one cell. That's mind-blowing.
It truly is. Cell signaling and communication are absolutely paramount. Cells have to precisely interact to make developmental decisions. A great example is the Drosophila neurogenic ectoderm. Here, cell-to-cell signaling, involving pathways like the Notch pathway, determines whether adjacent cells will become skin cells or neurons. It's a kind of local decision-making process.
Wow. And the precision of something like Drosophila embryogenesis overall is just stunning, isn't it?
Breathtaking. You start with maternal effect genes, like becoid, setting up an anterior gradient, basically telling the embryo which end is the head.
Like a chemical landmark.
Exactly. And nanos sets up a posterior gradient for the tail end. These gradients then control the expression of subsequent layers of genes, like the parallel genes. Even skipped, Eve is a famous one. It gets expressed in these incredibly precise stripes. 7 perfect stripes along the embryo, helping to define the segments of the fly larva. And then you have the Hox genes.
Ah, the master body plan genes.
That's right, an evolutionarily ancient and deeply conserved set of genes. They control the identity of different segments along the anterior-posterior axis, telling a segment whether it should be part of the head, thorax, or abdomen, for instance. And they show this amazing property called collinearity.
Meaning their order on the chromosome matches where they're expressed in the body.
Exactly. Their physical order along a chromosome mirrors the order of the body parts they specify from head to tail. And this is conserved across incredibly diverse species, from flies to humans. It's a fundamental genetic toolkit for building bodies.
And speaking of evolution, that's the grand narrative, right? How this blueprint changes over vast timescales.
Precisely. Things like gene duplication, where a gene gets accidentally copied.
You're reading a spare.
Yeah, creating a spare copy that's then free to diverge and potentially acquire a new function. That, combined with changes in those non-coding regulatory elements we talked about, the enhancers and promoters, drives evolutionary innovation. It allows life to constantly adapt and create new forms.
But sometimes evolution throws curveballs, right?
Yeah.
Like those anomalous genes.
Absolutely. The book mentions the cellulose synthase gene. Cellulose is the main component of plant cell walls, wood.
Definitely a plant thing.
You'd think. But this gene, or one very much like it, is found in the sea squirt, an animal. It highlights these really unexpected evolutionary paths, maybe through horizontal gene transfer or some other unusual mechanism. It's a fantastic reminder that evolution isn't always neat and tidy.
Nature's full of surprises.
Indeed. And if we connect this to the bigger picture, molecular biology is now moving beyond just studying individual genes in isolation. It's increasingly about understanding life as an integrated system.
Which brings us to systems biology.
Exactly. It's an emerging discipline, really. It combines traditional wet lab experimental biology with powerful computational analysis and modeling. The goal is to uncover the underlying design principles of complex cellular processes.
Design principles, like engineering.
In a way, yes. It looks at regulatory circuits, how genes and proteins interact in networks to control cellular behavior. For example, positive autoregulation, where a protein activates its own gene, can create bistable switches, like a light switch that's either fully on or fully off with no in-between state. This can lead to cellular memory, where a cell remembers a past event.
Wow. What about negative?
Negative autoregulation, where a protein inhibits its own gene, tends to make gene expression more robust. It reduces noise, those random fluctuations in protein levels, leading to more stable and predictable output.
Stabilizing the system.
Right. And then you have more complex motifs, like feed-forward loops. These often act like AND gates, requiring multiple inputs or conditions to be met before activating a target gene. Adds another layer of decision-making.
And oscillating circuits.
Yeah, those are fascinating. Genes that rhythmically turn on and off, often coordinated by signals between cells. We see this beautifully in the formation of body segments, the somites, during vertebrate development. It's like a biological clock ticking away to pattern the embryo.
This is where biology gets truly exciting, isn't it? When you see these sophisticated, almost engineering-like principles at work within living cells, creating these dynamic, adaptable systems.
It really is.
So what does all this mean for you, the listener? We've gone on quite a journey, haven't we? From the fundamental rules of inheritance Mendel figured out to the iconic double helix, that intricate dance of transcription and translation, the constant vital battles against mutation, and these incredibly complex networks that control every single gene, shaping life as we know it.
I think it means that the blueprint of life is far more dynamic, far more adaptable, and just exquisitely regulated than we probably ever imagined initially. It's really a testament to the elegant solutions evolution has found and continues to find for life's most fundamental processes.
Yeah, and that wraps up our deep dive today. What's fascinating here, I think, is just how much we've learned, especially in the last 70 years or so. Yet how many layers still remain to be uncovered about this incredible molecular world.
Indeed, and perhaps this raises an important question for you to ponder. If RNA was likely both the genetic material and the main catalyst in early life, the so-called RNA world, and given its ongoing, incredibly versatile roles today in things like splicing, catalysis in the ribosome, and all sorts of gene regulation, does its pervasive influence perhaps hint at a deeper, more fundamental purpose or potential that we are only just beginning to truly uncover?